In previous video, we discussed about receptor and neural function of retina. Today, we will discuss about phototransduction in detail. Now, what is meant by visual phototransduction? So, the entire process by which the light initiates a sensory response, it is called visual phototransduction. In other words, we can say that it is a process by which light is converted into electrical signals in the rod cells, cone cells, and photosensitive ganglion cells of the retina. To understand the phototransduction, it is important to understand role of different currents and how they alter the resting membrane potential in photoreceptors. Now, the resting membrane potential in other sensory receptor cells, it is usually between -70 to -90 millivolts. But photoreceptor cells, they are unusual because they depolarize to -40 millivolts in response to absence of stimuli or darkness, or we can say in scotopic conditions. This happens because in the dark, cyclic GMP levels are high and they keep cyclic GMP gated sodium channels open, thereby allowing a steady inward current. We call it the dark current. This in turn keeps the cell depolarized and maintains its resting membrane potential at about -40 millivolts. Now, when photon strikes the photoreceptors, this dark current is abolished and receptors are hyperpolarized and potential reaches -70 to -80 millivolts. When other sensory receptors are excited, the electrical response is in the form of depolarization, whereas in photoreceptors, the response is in the form of hyperpolarization. During scotopic conditions, when receptors are in depolarized state, they release neurotransmitter glutamate at a steady rate. This glutamate, though usually it is excitatory, but here in retina, it functions as an inhibitory neurotransmitter. Now, when the light falls on retina, these photoreceptors, they are hyperpolarized, and hyperpolarization in photoreceptors reduces the release of glutamate, which in turn reduces the inhibition of retinal nerves. And this leads to development of response in bipolar and ganglion cells, so that the action potentials are transmitted to cerebral cortex via the optic pathway. Now let's see how all these above events happen when light strikes the retinal photoreceptors. Retinal photoreceptors contain photosensitive pigment, which is rhodopsin in rod cells and photopsins in cone cells. We already discussed in previous videos that photopsins are of three types, that is porphyropsin, iodopsin and cyanopsin. All these pigments are basically combination of 11-cis retinal and opsin. 11-cis retinol, it is an aldehyde of vitamin A. When light photon interacts with 11-cis retinol in photoreceptor cells, it undergoes isomerization, changing from 11-cis to all trans configuration. The opsin therefore undergoes a conformational change to meta-rhodopsin 2. This meta-rhodopsin 2 in turn activates a G protein known as transducin. Now, transducin, it has three subunits. They are alpha, beta and gamma. Now, when this meta-rhodopsin 2, it activates transducin, this causes it to dissociate from its bond GDP and bind to GTP. Then the alpha subunit of transducin dissociates from beta and gamma subunits, with the GTP still bound to the alpha subunit. Now, the alpha subunit GTP complex, it activates cyclic GMP phosphodiesterase, also known as PDE6. This phosphodiesterase hydrolyzes cyclic GMP, forming 5' GMP. This in turn lowers the intracellular concentration of cyclic GMP, and therefore the cyclic GMP gated sodium channels they close, which in turn reduces the influx of both sodium and calcium ions. But sodium ions, they continue to be pumped outwards through a membrane of inner segment through sodium-potassium ATPase pumps. And calcium ions are excluded through rapid cation exchangers. Thus, more sodium ions now leave the rods than the sodium ions which are leaking back in. Because they are positive ions, their loss from inside the rods create increased negativity inside the membrane. And the greater the amount of light energy striking the rod cells, the greater the electronegativity becomes inside. That is, the greater is degree of hyperpolarization. At maximum light intensity, the membrane potential approaches -70 to -80 millivolts, which is near the equilibrium potential for potassium ions. Now, this hyperpolarization of cell causes voltage-gated calcium channels to close, further dropping the calcium level. As the calcium level in the photoreceptor cells drops, the amount of neurotransmitter glutamate that is released by the cells also drops. This is because calcium is required for glutamate containing vesicles to fuse with the cell membrane and thereby release their contents. A decrease in the amount of glutamate released by the photoreceptors leads to development of response in bipolar cells and ganglionic cells, so that the action potentials are transmitted to cerebral cortex via the optic pathway. Now, following image depicts different stages of phototransduction which we discussed just now. So you can pause this video here and look at this image. This image is self-explanatory. So let's continue further. Now at this point I want to discuss about a very important preventable condition of eye which develops due to vitamin A deficiency. This condition is night blindness, also known as nyctalopia. We already discussed before that retinal is an important component of photopigments in rods as well as cones. So in case of severe vitamin A deficiency, the amount of retinal and rhodopsin that can be formed are severely depressed. Vitamin A deficiency also contributes to blindness by causing the eye to become very dry, which in turn damages the cornea and retina. Vitamin A deficiency, it first alters the rod function, but if the deficiency becomes more severe, ultimately the cone degeneration also occurs. Prolonged deficiency of vitamin A is associated with anatomic changes in rods and cones, followed by degeneration of neural layers of retina. For night blindness to occur, a person usually must remain on vitamin A deficient diet for months together, because large quantity of vitamin A is normally stored in the liver and can be made available to the eyes. So vitamin deficiency, if it prolongs for months together, then only the person will develop night blindness. The treatment of vitamin A deficiency is simple. Just we need to give vitamin A supplements. But treatment with vitamin A can restore retinal function only if given before the receptors are destroyed. Now these two images here shows comparison between normal vision and the vision of a night blind person. Now let's discuss how this phototransduction cascade is deactivated. This is very important because the photoreceptor's response to a single photon must be terminated so that the cell can respond to another photon. We already discussed that in light, low cyclic GMP levels close the sodium and calcium channels, reducing intracellular sodium and calcium. These lower calcium levels indeed help in termination of phototransduction cascade by multiple mechanisms. The lower intracellular calcium level, it causes calcium to dissociate from guanylate cyclase activating protein GCAP, which then interacts with guanylate cyclase and activates it. Now activated guanylate cyclase then proceeds to transform GTP to cyclic GMP, which in turn restores depleted cyclic GMP levels and thus reopens the cyclic GMP gated cation channels, thereby restoring the dark current. Low intracellular calcium level also causes calcium to dissociate from GTPase activating protein GAP or GAP. The liberated GTPase activating protein then deactivates transducin, terminating the phototransduction cascade and thereby restoring dark current. Lower intracellular calcium level also makes intracellular calcium recoverin rhodopsin kinase complex to dissociate into calcium and recoverin and rhodopsin kinase. The liberated rhodopsin kinase then phosphorylates the metarhodopsin 2, thereby reducing its binding affinity for transducin. Finally, another protein arrestin then completely deactivates the phosphorylated metarhodopsin 2, effectively terminating the phototransduction cascade and thereby restoring dark current. Finally, low intracellular calcium level makes the calcium calmodulin complex within the cyclic GMP gated cation channels more sensitive to low cyclic GMP levels, thereby keeping the cyclic GMP gated cation channels open even at low cyclic GMP levels and thus restoring the dark current. Now finally, let's briefly discuss about light and dark adaptation. If a person spends a considerable length of time in brightly lit surroundings and then moves to a dimly lit environment, the retina slowly becomes more sensitive to light as the individual becomes accustomed to the dark. This decline in visual threshold is known as dark adaptation. It is nearly maximum in about 20 minutes, although some further decline occurs over a longer period of up to 2 hours. On the other hand, when one passes suddenly from a dim lit to a brightly lit environment, the light initially seems very intense and even uncomfortably bright until the eyes adapt to the increased illumination and the visual threshold rises. This adaptation occurs over a period of about 5 minutes and is called as light adaptation. There are 3 important processes which are involved in light and dark adaptation. First is that the photochemical concentration decreases as it is broken down into retinal and opsins when exposed to bright light, thereby decreasing the sensitivity of the eye to the light. Whereas in dark, the photochemicals are replenished back, thereby increasing the sensitivity of the eye to light. The adaptation by photochemical system is a slow process and it takes minutes to hours. But this is the most effective adaptation process. The other two adaptation processes, they are neural adaptation and adaptation by altering the pupillary diameter. These latter two processes are very rapid and occurs within fraction of seconds. But compared to photochemical system, they are less effective. Now let's come to neural adaptation. So the neural adaptation involves neurons in the successive stages of visual chain in the retina and in the brain. Here, when light intensity first increases, the signals transmitted by the bipolar cells, horizontal cells, amacrine cells and ganglion cells are all intense. However, most of these signals decreases rapidly at different stages of transmission in the neuronal circuit, thereby adapting to bright light. Now finally, let's come to adaptation by changing the pupillary size. So in bright light, the pupil constricts, thereby effectively reducing the amount of light which is entering the eye. Whereas in dark, the pupil dilates, thereby allowing more light to enter into the eyes. So that's all for today. In next video, we will discuss about image forming mechanisms and various defects which are associated with them. So keep watching. Thank you. Have a nice day.